Hello students, in this part we are going to discuss about the conduction of nerve impulse or the mechanism of conduction of nerve impulse. So the first step that we are starting with is the generation of nerve impulse. Now let us see how the nerve impulse is generated. Now when this nerve fiber on its resting position, or when the nerve fiber is in its resting state, at that time we have seen that outer side of the nerve fiber or the outer side of the membrane, nervous membrane or neurolemma, the outer side is electropositive and the inner side is electronegative, and these charges are because of the sodium ions which are present in more quantity outside the neurolemma or outside the nervous membrane. The concentration of sodium ions is more, and the positive charge which is there on the outer side of the membrane, that is because of the sodium ion concentration, more sodium ion concentration. Whereas the inner side of the membrane is electronegative, and this is because of the negative proteins which are present in the inner side of the membrane, in the neuroplasm. In the neuroplasm, the negative proteins are present, and because of these negative proteins, the negative charge is the inner side of the membrane gets its electronegative charge, whereas outer side in the extracellular fluid there are more concentration of sodium ions, and because of this more concentration of sodium ions, the charge of the membrane on the outer side is electropositive. This is at the resting state. At this resting state, these charges which are acquired by the membrane, positive on the outer side, negative on the inner side. So these charges are acquired on the membrane, and hence this membrane is said to be polarized. When the outer side of the membrane is electropositive and the inner side is electronegative, at this state the membrane is said to be polarized. Now this is also called as the resting state of the nerve fiber or resting position of the nerve fiber. Now here at the resting state, the potential of the nerve fiber, that is also called as the resting potential. The potential of the nerve fiber is minus 70 millivolts. Now here at this potential, the sodium channels are closed. Now if we have to draw the sodium channels in a diagrammatic representation, it would be like this. These are the sodium channels or sodium voltage gates, we can say. This is the activation gate, this is the inactivation gate. This is sodium channel. Now here it is closed because the nervous membrane or the nerve fiber is at the resting potential or the resting state. That's why the sodium ions or sodium channel gates, those are closed. Here these are the sodium channel gates. This is the nervous membrane. Outside the membrane, in the extracellular fluid, there are sodium ions, more concentration of sodium ions. Whereas inside the membrane, in the neuroplasm, there are potassium ions. So, because of these sodium ions, the outer side of the membrane is electropositive, whereas the inner side of the membrane is electronegative because of the negative proteins which are present in the neuroplasm. So here the outer side is electropositive and the inner side is electronegative because of the ions and the proteins which are present in the extracellular fluid. In extracellular fluid there is more concentration of sodium ions, so therefore the outer side of the membrane is electropositive, whereas the inner side has more negative proteins, and that's why the inner side of the membrane is electronegative. Now here these channels, these sodium channels, they open because of the voltage. When the stimulus acts on the nerve fiber, when the nerve fiber gets the stimulus, at that time a voltage is generated. impulse is generated, voltage is created, and because of that voltage, the sodium channels, they open. Now, if we have to represent it in a diagrammatic form, it would look like this. This sodium ions, when the stimulus is generated or when the stimulus is given to the nerve fiber, at that time what happens? The resting potential, which was minus 70 millivolts, it changes to minus 55 millivolts. The resting potential changes into action potential. So it becomes minus 55 millivolts. And at that time, when this resting potential changes into action potential, at that time the sodium channel gates, they open. And when these sodium channel gates open, the sodium ions which are present in the extracellular fluid, they start moving inside the neuroplasm. These sodium ions, they start moving inside the neuroplasm. Now, if we have to show this in this line diagram, it would look like this. Suppose this is the membrane. This is the membrane, nervous membrane, neurilemma, and outside it, before we saw, outside it electropositive charge is there. Inside the membrane is electronegative charge. Now, when these sodium ions, they move inside the neuroplasm, at that time what happens? Suppose here are the sodium ions. When the stimulus is acting on the nerve fiber, at that time when the nerve fiber receives the stimulus, at that time these sodium ions, they move inside the neuroplasm with the help of these sodium channel gates or voltage gates of sodium. With the help of these sodium ions, they move inside the neuroplasm. And after they move inside the neuroplasm, what happens? Here the membrane was said to be polarized. Outside was positive charge, inside was negative charge. But after the sodium ions enter inside the neuroplasm, what happens? These charges, they change. The outer side becomes electronegative and the inner side becomes electropositive. This is said to be the generation of impulse. Here we say that the impulse has been generated. Because of the sodium ions moving in, the negative charge which was inside the membrane changed into positive charge. And the positive charge was outside the membrane, it changed into negative charge. At this state, we say that the nerve membrane or the neurilemma is depolarized. Here, all the charges were same. Outside was positive, inside was negative, everywhere. But here now, the impulse has been generated. The charges have changed. So, this polarized membrane becomes here depolarized. So, it is said that the membrane has been depolarized. When the charges change, the outer one becomes electronegative and the inner one becomes electropositive. We say that the impulse has been generated. And we also say that the membrane has been depolarized. Now here, this generation of impulse can be shown in a graphical way. Here, suppose this is the graph of time against the millivolts or against the voltage. Now here, suppose this is at resting potential, resting position of the nerve membrane, that is minus 70 millivolts. Minus 17 millivolts, we said that it is a resting potential, means the nerve fiber is at its resting state. What happens when the stimulus is received? When the nerve fiber receives the stimulus, at that time this resting potential, it changes into action potential. It changes to minus 55 millivolts. At this time, this graph, it slightly goes up. It means the impulse is being generated. The channel gates, they open. The channel gates open. They allow the sodium ions to move inside the neuroplasm. Now, when it comes to zero, at that time what happens? When it moves further above zero, it becomes positive. Here it was negative, minus 70 and minus 55. It becomes positive. Till from 0 till it comes to plus 45. At that time, in between this time, what happens? These sodium ion gates or sodium channel gates which are there, they start closing slowly. They start to close slowly, and at plus 45, all the sodium channel gates, they close down, and the potassium channel gates which are there, they open. And here it is said that the action potential is generated. At 55 we said that the action potential was generated. Now from plus 45 to plus 50, this action potential works. The sodium ions, they open slowly. All the sodium ions, sodium ions gates, they open, and sodium ions which are present outside the membrane in the extracellular fluid, they start moving inside the neuroplasm. And from this plus 45 to 50, at that time what happens? The sodium ions, they slowly close down, and sodium ions, they get closed, and the potassium channels, they open. Now these potassium channels, they open to pump the potassium ions outside. As this sodium gates, they allow the sodium ions to move inside. The potassium gates are there, they allow or they pump the potassium ions outside to balance the charge. Now here, as the sodium ions, they move inside, in the inner side will be electropositive, the outer side will be electronegative. To balance this gradient, electron gradient or charge gradient, to balance this concentration gradient, the sodium channel gates, they open, and they pump the potassium channel gates, they open, and they pump the potassium ions outside to balance the charges, to balance the gradient. Now these sodium ions, they enter inside the neuroplasm because of the gradient, because outside it was positive charge, inside it was negative charge. To balance these, the sodium ions, they come in because of the concentration gradient. Now as the sodium ions, they come in, the inner side will be electropositive, the outer side will be electronegative. To balance these charges, the sodium channels, they close down, and the potassium channels, they open. And these potassium channels, they pump the potassium ions outside the membrane to balance the charges, to balance the gradient. So this is how the impulse is generated. When the nerve fiber is in its resting state, it has electropositive charge outside, and it has electronegative charge inside. The positive charge is because of the sodium ions which is present outside the membrane. Negative charge is because of the negative proteins which are present inside the membrane. Now this state of the nerve fiber is said to be the resting state or resting position of the nerve fiber. And this state, the electropositive charge is outside, electronegative charge is inside. This state is said to be the polarized state. In this state, the nerve fiber is said to be polarized. At this state, the resting potential of the nerve fiber is minus 70 millivolts. And here, when the impulse is given to the nerve fiber, when the stimulus is given to the nerve fiber, at that time the resting potential changes into from minus 70 to minus 55. Here we say that the resting potential changes into the action potential, or we say that the action potential is generated. Now as the resting potential changes to minus 55 millivolts, it opens the sodium channel gates. As the sodium channel gates open, the sodium ions which are present outside, they start to move inside the neuroplasm. As these sodium ions, now why these sodium ions, they move inside? Because of the potential gradient, because of the concentration gradient. Here outside in the extracellular fluid, the concentration of sodium ions is more as compared to the neuroplasm. So these we have learned that the particles, they move from higher concentration to lower concentration. So here sodium ions, they move from the extracellular fluid, from outside the membrane to inside the membrane in the neuroplasm. Now, as these sodium ions, they move in, the inner side of the membrane becomes electropositive because of the sodium ions, and the outer side becomes electronegative. Here, it is said that the impulse has been generated. The impulse has been generated and the membrane becomes depolarized. Here, it was in polarized state. Here, it becomes depolarized because the charge is changed. And it is shown in a graphical representation. At minus 70, the nerve fiber is at its resting position, resting potential, that is minus 70 millivolts. When the stimulus is given to the nerve fiber, the resting potential changes into action potential at minus 55 millivolts. Now from this, the graph rises, or this line rises to plus 45. Here the sodium channel gates, they close down, and potassium channel gates, they open to pump out the potassium ions. Now, how this the conduction of this nerve impulse is taking place, that we will see in the next